Okay, Assalamualaikum everyone. So today we are going to look at the second part of chapter 1. In the previous session we have already went through a brief history of the cytology. We have looked at the working principle of the light microscope and also about the electromagnetic spectrum. So today we are going to proceed with the electron microscope. Remember in the previous session we have discussed that the resolution of the light microscope is only down to 200 nanometers. Therefore, the light microscope can only see structure or organelles that has a size that is larger than 200 nanometers. And the scientists thought that they can never see any objects that is smaller than that. So they tried to build a microscope using radiation of shorter wavelength. They tried to work with UV ray, they tried to work with X-rays, but both turned out to be too difficult to focus onto the specimen. So they finally discovered that electron was the best option. Not only because electrons have extremely short wavelength, they are also chosen because of the negative charge that they have, and this allows scientists to use electromagnets to focus them easily in the electron microscope that they were going to build. So the working principle of the electron microscope is a bit different from light microscope. In light microscope, the source of radiation is the visible light, while in the electron microscope, like what I have said just now, the source of radiation will be the electrons. So in this diagram, you can see that unlike the light microscope, where the source of light comes from the base, the position for the source of electron is on top, and it is produced by a structure known as the electron gun. Once the gun is given enough energy or heat, the electron inside of the electron gun will excite to a higher energy level and thus able to escape their orbit and therefore escape the metal gun as well. And free electrons behave just like electromagnetic radiation with much shorter wavelength, and they can easily be focused using electromagnetic lenses. So all this cylindrical shape structure that you see here are the lenses, or sometimes known as the positively charged cathode. So the specimen is positioned here, and the beam of electrons will hit and penetrate the specimen at different speed. If the electrons hit the specimen in the middle, for example, and if it just happened that the middle part of the specimen is denser than the rest, so the penetrated electrons will pass through with slightly reduced speed. The beam will finally be focused onto a fluorescent screen down here. The areas hit by electrons will shine brightly, producing a black and white picture. However, electrons that pass through a denser part of the specimen will hit the fluorescent screen at much slower speed and produce a much darker image. So the contrast of this dark and light color on the fluorescent screen will produce an overall image of the specimen. So these are just some of the points that I've mentioned earlier. You can copy it down if you want to. Talking about the image that is produced by the microscope, there are actually two different types of image based on the type of electron microscope used. The first image is known as a transmission electron micrograph, or in short known as the TEM. In transmission microscope, the beam of electrons are passed through the specimen before being viewed. So this microscope will allow you to see thin sections of the specimens because only those electrons that are transmitted are seen, right? Only those electrons that manage to pass through the specimen will be viewed. And this is actually the most commonly used electron microscope and was the first one to actually be created. And this is how the image will look like. Darker area of the transmission electron micrograph means that the area is denser, while lighter area of the transmission electron micrograph means that the area is thinner or less dense. Second type of image is known as the scanning electron micrograph, or in short, it is known as SEM. In scanning electron microscope, the electron beam is used to scan the surface of the structure. And unlike TEM, or unlike transmission electron micrograph, in scanning electron micrograph, only reflected beam is observed. In scanning electron micrograph, the electrons are reflected at various angles, which will affect the size of the wavelength. Okay, therefore, reflected electrons with a longer wavelength will produce a darker image, while reflected electron with shorter wavelength will produce a lighter image. Okay, which is the same concept used in transmission electron microscope. And these reflected electrons will produce a three-dimensional appearance of the specimen. However, because most of the electrons' wavelength are affected, the resolution of SEM, or scanning electron microscope, is usually lower than the transmission electron microscope, which is about 3 nanometers to 20 nanometers, as compared to transmission electron microscope that has a resolution of 0.5 nanometers. This is how the image will look like. Darker area of the picture, or darker area of the scanning electron micrograph, means that the degree of reflection is bigger and therefore will have longer wavelength, while lighter area of the picture or the scanning electron micrograph means that the degree of reflection is smaller and therefore will have a shorter wavelength. Next, we are going to look at the structures and organelles in the cell. You might have already learned some of the organelles before, and in the A-level syllabus, we are going to look at a few more. So we will start with the ultrastructure of the animal cell. And in this general diagram, okay, it shows you those organelle and structures that you need to know, and how they look like. You have the largest one in the middle, known as the nucleus, and inside the nucleus there are the genetic component known as the chromatin or chromosome, and there is also a nucleolus here. And just outside the nucleus, okay, you can see that there is a structure known as the rough endoplasmic reticulum. A bit further towards the edge of the cells are the smooth endoplasmic reticulum and also the Golgi body. You can also see a lot of ribosomes attached to the rough endoplasmic reticulum, and also suspended inside the cytoplasm are centriole, lysosome, and mitochondria. So we are going to look at all of these structures in much detail after this. And this is a transmission electron micrograph of the animal cell, okay, as seen through a transmission electron microscope. Okay, so a few very obvious structure that you can see here. You can see the nucleus, Okay, the large circular structure in the middle of the cell is the nucleus, and all this blue structure within the cytoplasm are the mitochondria. And you can also see some endoplasmic reticulum here and there. Okay. The first organelle that we are going to look at is the nucleus, which is the largest organelle inside the cell. It is surrounded by two layers of membrane, okay, with small pores known as the nuclear pore. Okay, the pores allow and control exchange of material between the nucleus and the cytoplasm. And if you look closely, you will notice that the outer membrane of the nucleus is actually continuous with the membrane of the rough endoplasmic reticulum. Okay, and inside the nucleus, you can also find the genetic makeup of the organism known as chromatin or chromosome. The chromatin is made up of DNA, which is organized into functional units known as genes, okay, which is inherited from one generation to another. One of the functions of the genes is that it holds information on enzyme synthesis. And because enzymes control almost all biochemical reaction of the cell, they are said to control all the activity of the cell as well. That's why, when you learn in IGCSE, when you learn biology in IGCSE, you were told that the function of the nucleus is just that, right? It's just to control the activity of the cell. Anyhow, we are going to learn about it later in the next chapter. So these are some of the points that I've mentioned about the nucleus. Next is the endoplasmic reticulum. It is a structure made up of a single membrane, and the membrane will form an extended system of flattened compartments or sacs spreading throughout the cell, right? Okay, just like what you are seeing here. And like I said just now, the membrane is continuous with the outer membrane of the nucleus. Okay, that's why you can see that the RER will always be positioned or arranged just beside the nucleus. Two types of ER are two types of endoplasmic reticulum, the rough endoplasmic reticulum and the smooth endoplasmic reticulum. Rough endoplasmic reticulum is so called because it is covered by numerous tiny organelles known as the ribosomes, okay, which give it the rough appearance. Okay, and ribosomes, maybe you have learned it before, are the site for protein synthesis, and you can find them attached to the RER or to the rough endoplasmic reticulum, or they can also be found suspended freely in the cytoplasm, right? So there are two types of ribosomes, the one that is attached to the rough endoplasmic reticulum, or the one that is suspended freely in the cytoplasm. The function of the ribosome is to synthesize protein, and protein that has been synthesized will move into the sacs of the rough endoplasmic reticulum to be transported in small vesicles, okay, to another organelle known as the Golgi body. Why? Because proteins made by the ribosomes are what we call as raw protein, okay, and they are a non-functional protein. And to turn them into a functional protein, okay, like enzyme or antibodies, they must first be modified, and the modification process takes place in the Golgi body. And for smooth ER, okay, it basically has a very similar structure as the rough ER or rough endoplasmic reticulum, except without the presence of the ribosomes. And the function of the smooth endoplasmic reticulum is to synthesize lipids such as cholesterol, which is a compound needed in the formation of plasma membrane, or to synthesize steroids such as estrogen or testosterone, which are the hormones in the human reproductive system. Some of the points about the endoplasmic reticulum, okay, that I've mentioned earlier. So next organelle that we are going to look at is the Golgi body. Basically, it has similar structure with the endoplasmic reticulum, which is a stack of flattened sacs. Okay, they are said to formed from vesicles which bud off from the endoplasmic reticulum. So whenever a protein has been made by ribosomes on the rough endoplasmic reticulum, the protein will move into the rough endoplasmic reticulum and travels to the edge of the rough endoplasmic reticulum to be packed in small vesicles that bud off from the membrane of the rough endoplasmic reticulum itself. And like what I have said just now, the function of the Golgi body is to collect, to process, and also to modify molecules, especially protein, to be transported out of the cell or to be used within the cell itself. If the protein is intended to be used by the cell itself, okay, the protein containing vesicles that bud off from the Golgi body will form an organelle known as lysosome. Okay, we are going to look at lysosome after this. But if the protein is intended to be used out of the cell, the Golgi body will pack the modified protein in small vesicles to be secreted out of the cell. And these vesicles are known as the secretory vesicles. Okay, the modified protein in the secretory vesicles can be enzymes such as maltase, protease, lipase, etc. Some points on Golgi body or Golgi apparatus that we have discussed just now. Next is the lysosome, and we have already learned just now that lysosomes are structures that comes from vesicles that bud off from the Golgi body. So they have modified protein inside of them as well, and most of the modified proteins are enzyme. And to be precise, it's hydrolytic enzyme, which is used to break down substances and compounds. And because of that, the function of lysosomes are to digest and break down unwanted structure like old and worn out organelles. In terms of structure, the lysosomes are spherical in shape with a size of 0.1 to 0.5 micrometers and are contained by a single membrane. Okay, some points on lysosome. Next organelle is the mitochondria. Most of mitochondria has the size of roughly about 1 micrometer in diameter and can vary in shape. Okay, but the most common shape for mitochondria is the sausage-like structure or the bean-like structure. The mitochondria is surrounded by a two-layer membrane. The inner folded membrane is known as cristae, while the interior solution is known as the matrix. The space between the inner and outer membrane is known as the intermembrane space. Okay, the number of mitochondria in a cell varies depending on the function of the cell. An actively working cell like the muscle cell may have up to roughly 1000 mitochondria to produce sufficient supply of energy. When we talk about the function of mitochondria, basically it is to generate energy, okay, from energy-rich molecules like sugars and fats. And the energy is generated in the form of a molecule known as adenosine triphosphate, or ATP in short. I will not talk into details about how mitochondria generates this ATP because you are going to learn this in the first chapter of your A2. But in general, the reaction is known as cellular respiration and occur in 4 different stages, okay, namely glycolysis, link reaction, Krebs cycle, and oxidative phosphorylation. Some of the reactions occur in the matrix, and some of the reactions occur on the cristae, and that is why the inner membrane is folded. Okay, that is why you have the folded inner membrane known as the cristae, because it gives a bigger surface area for the 4 reactions that I've mentioned just now. It was also discovered back in the 1960s that mitochondria and chloroplasts contain small circular DNA molecules, just like those found in bacteria. And it was later proved that mitochondria and chloroplasts are in fact ancient bacteria which now live inside larger cells, like cells of the animals and cells of the plants. And this is what we call as the endosymbiont theory. Some of the points that I've mentioned earlier about the mitochondria, and like I said, don't worry too much if there are certain points here that you just can't figure out or imagine. For example, number 2 here points out that the reaction uses hydrogen and electrons, and you will understand why and how when you learn chapter 12. Everything will become crystal clear 1 year from now, or if you want to understand it sooner, you can just flip your textbook and go through the whole chapter 12. Next is the plasma membrane or the cell surface membrane. Not much is described now because you are going to have a specific chapter about it in chapter 4. But for now, it is good enough for you to know that plasma membrane or cell surface membrane is the structure that surrounds the cell or the organelles. It is partially permeable and therefore it controls the exchange of materials between the cell and its environment. Here's some of the points on plasma membrane. Next, we have a structure known as the microtubule. Microtubule is basically a long, rigid, hollow tube found in the cytoplasm. They make up the cytoskeleton that provides structural support and gives shape to the cell. Very similar in terms of function like the skeleton or bones that we have in our body, right? So if you look in the diagram, you can see that the microtubule is made up of 2 types of protein known as the alpha tubulin and beta tubulin. The alpha and beta combine to form a dimer. A dimer is actually a double molecule, right? A double molecule like you see here. And the dimers are then joined end to end to form a long structure known as the protofilament. Okay, like what you see here. Okay, 13 filaments will line up side by side in a ring formation to form a cylindrical structure with a hollow center, okay, like what you see in the illustration here, and also the transmission electron micrograph on the right side of the diagram. Okay, some of the points on microtubules that I've mentioned just now. And this is another diagram to show how the microtubules are arranged, okay, from their alpha and beta tubulin units. With proper staining of the plant specimen and proper technique, we can actually see through the microscope the extensive network of the cytoskeleton made from the microtubule in providing support and structure to the cell. Next organelle is the centrioles and the centrosomes. Centrioles are organelle that is located just outside the nucleus. Okay, centrioles usually exist in pairs and lie close together at a right angle to each other in a region known as centrosome. Okay, in terms of structure, centriole is hollow and cylindrical with the length of about 500 nanometers. Okay, they are formed from 9 triplets of microtubule arranged in a ring, okay, just like what you see here, okay, in this transmission electron micrograph. Okay, and the function of centriole is as microtubule organizing center, okay, or MTOC. We're going to look at this in much detail when we learn chapter 5, okay, that assembles new microtubule. And they are also believed to produce spindle fiber during mitosis or meiosis. And we'll also discuss this further when we learn, or when we discuss chapter 5 later on. So the diagram that you see here give a better illustration on how centrioles are arranged. You have 9 triplets of microtubules here, and they are arranged, okay, in a ring formation that produce a hollow cylindrical structure. Okay, these are some of the points on centrioles that we have discussed just now. Next is the structure of cilia, which may be found in certain cells. Right, it's a small extension on the cell surface membrane or on the plasma membrane, and a longer version of the cilia is known as flagella. Right, so the short one is known as cilia. Okay, a larger extension will be known as flagella, and normally you can find them in unicellular organism such as Paramecium, Chlamydomonas, where they are used as a locomotion mechanism. And the cilia and flagella are also believed to be made up of microtubules. And then certain cells like those in the small intestine will also have finger-like extension, okay, from the cell surface membrane, and they are known as the villi or microvilli. They increase the surface area of cells in the small intestine for an efficient absorption of the nutrients. And the last organelle that we are going to look at in this chapter is the chloroplast. Okay, in terms of structure, the chloroplasts have an elongated shape. The size of the diameter of the chloroplast is about 3 to 10 micrometer, which is relatively larger compared to the mitochondria. Right, so that's why if you look at the plant cell under the light microscope, okay, the chloroplast can easily be observed. okay, as compared to the mitochondria. Okay, just like the mitochondria, okay, chloroplast is also surrounded by two membranes forming the chloroplast envelope. The inner membrane projects inside to form a fluid-filled sacs called thylakoids, okay, which spread out like sheets in 3 dimensions, right? So all these projections that you see here are thylakoids. While thylakoids, okay, form flat disc-like structures that stack up like piles of coins, many layers deep, okay, forming what we call as grana, right? So the thylakoid disc, okay, if it is a single disc, then we call it as thylakoids. But if the thylakoids stack up, okay, on top of one another, okay, we call them as grana. Okay, and the inner fluid that surrounds the grana is known as the stroma. Okay, so those are the things that you need to memorize. And this is a transmission electron micrograph of the chloroplast. So you can clearly see the extension of the inner membrane that produces the thylakoid and the grana. Some of the point that I have discussed earlier, okay, about chloroplasts. Okay, so I don't want to bother you too much on the function of chloroplast because it's difficult to imagine all of the points listed here, because just like mitochondria, there will be a specific topic dedicated to discuss the function of chloroplasts in A2. However, in general, we can say that the function is to carry out photosynthesis, where light energy from the sun is converted into chemical potential energy, such as the inside the glucose molecule. And glucose that have been synthesized will immediately converted to sucrose and starch to be stored. And just like mitochondria, okay, the endosymbiont theory also stated that chloroplasts originated from a blue-green bacteria, and this explains why the chloroplasts have their own circular DNA and ribosome, because only bacteria have them. And when you talk about cells, besides classifying it into animal and plant cells, we can also classify them into two fundamentally different types. The first one is known as the prokaryote. And there are cells that are simpler in forms, and the most common feature of a prokaryote is that they don't have any nucleus and most membrane-bound cells. Their DNA lies suspended in the cytoplasm and sometimes accompanied by a circular DNA known as plasmid. They also have freely suspended ribosomes, which are much smaller than cells with nucleus. In terms of overall size, they can be down to 1 or 10,000 times smaller than cells with nucleus. An example of prokaryotes are the bacteria. The second type of cell is the eukaryote. Basically, they are cells with nucleus and membrane-bound organelles. Eukaryotes include animal, plant, fungi, and protoctis. They are said as the modern cells as they evolved about roughly 1.5 billion years after the prokaryotes. some points on the prokaryote and eukaryotes that I've mentioned just now. So these are a general structure of prokaryotes. On the left are the additional structures that can sometimes be found in certain bacteria, while on the right are structures that will always be present in all prokaryotes or bacteria. Okay, in this table, we have a list of differences between prokaryotes and eukaryotes. Basically, whatever features that a prokaryote have, it will be the opposite of the eukaryotic cells. And finally, a very brief description about viruses. So what is a virus? A virus is a small infectious agent that replicates only inside the living cells of an organism. Viruses can infect all types of life forms, from animals, plants, microorganisms, including bacteria. Uh, Viruses are considered by some to be a life form because they carry genetic material, they reproduce, and they evolve through natural selection, but they lack key characteristics that are generally considered necessary to count them as life. But because they possess some, but not all, such qualities, viruses have been described as organisms at the edge of life. Okay, and the general structure of virus is very simple. Okay, it has the outer capsid, okay, that protects the genetic material inside, and the genetic material can be made out of DNA or RNA. So this is an electron micrograph of how a bacteriophage attaches itself to a bacterial host before injecting its genetic material. And these are a list of some features of the virus. Okay, for example, number one, they have a very symmetrical shape. Protein coat is made up of separate protein molecules, each of which is called a capsomere, and the size is very small. Okay, it ranges about 20 to 300 nanometers. And all viruses are parasite, okay, because they can only reproduce by infecting and taking over living cells. Okay, and like I said just now, it contains DNA or RNA that will take over the protein synthesizing machinery of the host cell, okay, for them to replicate. Okay, and I guess with that we have concluded chapter 1. Okay, and inshallah I will see you again in the next session for the second chapter of your AS. Okay, with that, Assalamualaikum warahmatullahi wabarakatuh.